I color-coded this picture. See if this makes any sense. What we're looking at here in the lumen of the intestine are the fat globules being emulsified into smaller droplets. That's the job of bile salts. I color-coded the bile salts green, and then the fats yellow. When it comes to the point that the fats are digested by the pancreatic lipase, what happens is those fats can leave the micelle and just use simple diffusion to get into the simple columnar cell of villus. And once inside, those fats are processed by the cell. They're processed by the Golgi apparatus, and as they move through the Golgi apparatus will put a coating of protein on that fat. That's what I'm indicating with the pink color. This complex is called a chylomicron. So chylomicrons are produced within the Golgi. It's just the Golgi putting a protein coating, so to speak, around the fat. And then the Golgi does what it always does, right? It's going to package the chylomicrons in a membranous vesicle. So that black line that I'm drawing is a membranous vesicle. And then it would eject the chylomicrons from the cell using exocytosis. So once the chylomicrons are in the interstitial fluid, here's the thing, because they have that protein coating, they can't get into the blood. They're too big. And so what the chylomicrons will do, just like other proteins, is they'll be accepted by the lymphatic capillary. So bigger picture, we're saying that once the fats are digested out here in the lumen, those fats will pass into the simple columnar epithelial tissue by simple diffusion, get a protein coat, it's known as a chylomicron, get ejected into the interstitial fluid, and then get picked up by the lacteal, the lymphatic capillary. And so fats don't get absorbed into our blood, they get absorbed into the lymphatic system. So remember, the lymphatic system is a series of lymphatic vessels that are interconnected, lymph nodes that clean out the lymph, and these lymphatic vessels all empty the lymph back into the blood at 2 points. So the lymph that contains the chylomicrons gets emptied back into the blood at the right lymphatic duct into the right subclavian vein, and by the thoracic duct into the left subclavian vein. And so the chylomicrons do become part of the blood at the level of the brachiocephalic veins, going into the superior vena cava, going into the heart. In this way, we don't send 100% of the fat that we've absorbed to the liver. We avoid the liver and the hepatic portal system by having the fats absorbed into the lymphatic system and then emptied into the blood. So let's think back to the bile salts. What I'm doing in this picture is I'm color coding bile salts in green. So the bile salts will also passively enter the simple columnar by simple diffusion, and the bile salts will move into the interstitial fluid, but the bile salts are absorbed by the blood, not the lacteal. And so once the bile salts enter into the blood, they're on their way to the liver. So using this picture to summarize, bile salts enter into the duodenum with the bile. Bile salts emulsify the fat. And once that fat is absorbed into the lymphatic system, the bile salts will also get absorbed, but they get absorbed into the hepatic portal vein, into the blood supply. And that hepatic portal vein takes the bile salts back to the liver, and the liver will filter these bile salts out of circulation and recycle them back into the bile.